Hello, everyone. How are you doing? Welcome to the season finale. Yay! Yes, welcome to the sixth and final session, Steam Forward in collaboration with Southwest Africa. Wow, it has been an exciting journey, and I hope that you have enjoyed the ride. Let me just start by just saying thank you to Dr. Janita Pritchett. You've been amazing, you've been inspirational, it has been exciting, and we've been learning quite a lot. I'm sure the parents, together with the students that are here with us, would really back me up on this one. Thank you. If you had missed any previous sessions, please do go to our YouTube page and check it out. We have them there stored up for you, and you can view them at your own time and pace, and enjoy that which we have to offer. For today, we're going to be learning how to extract DNA from strawberry, and I guess it's going to be exciting. I'm looking forward to it. Dr. Janita, good morning, and you can take us. You can take over.
Thank you so much, Freddie, and thank you so much to the entire SciFest team for inviting me to share this space with you all. It's been quite a journey. We've had a lot of fun learning and getting our hands into some really cool science experiments. So if you've been on this journey with us for the last 6 weeks, we've been exploring different aspects of STEM and doing a lot of really cool science experiments along the way. So just to recap where we've been, right? So we started by learning how to blow up balloons with chemistry. So we use really simple acid and base reactions in order to blow up a balloon and to create lava lamps. The second week, we actually learned how to make our very own ice cream and explored the idea of physical changes and how we can use that to create ice cream or a dessert. The third week, we learned how to float an egg and explored all about density. The fourth week, we learned how to make chromatography, butterflies, and flowers. So we added a little art. So we went from STEM to STEAM. And then last week, we learned all about microscopes, and I got to show you guys some really cool things under our digital microscope. So for today, the finale, which I'm so sad about, I can't believe it's coming to an end. We're gonna be learning all about DNA and how to extract it from strawberries, okay? So DNA is the short part, the short word, or the acronym that we use. But what DNA stands for is deoxyribonucleic acid, okay? And DNA is really, really important. It's where we get our hereditary information, our traits. That's what makes all of us unique. It's present in every single cell, both plant and animal. And most of it is found inside the nucleus in the cell. And so because it's in the nucleus, we're going to be using some different materials to be able to extract it and take it out. So what does DNA look like? Well, obviously I've colored these and drawn them, but what DNA is, it's this double helix, these two strands of material that form this helical structure. And it's made of this it's a large molecule made of these smaller units, which are called nucleotides. Nucleotides have three components. They have a sugar component, which is typically the ribose component, a phosphate, and a nitrogenous base. Now, there are a couple different bases that exist. There's 4 main ones. And so there's adenine, or my yellow, thymine, or the green, guanine, or the red, and cytosine for the orange. And these different base pairs, they're able to match up and align with different nitrogenous bases in order to create this helical formation, okay? So we're not gonna see this exact helical thing because we really need a microscope to see that. But what we are gonna see are these long fibers that represent the DNA that we're able to extract. So why are we using strawberries, right? Strawberries are a fruit, they're great to eat. Well, strawberries actually have their chromosomes found in what's called an octoploid rather than a diploid. So in human cells, we have diploid chromosomes, which means there's 2 sets. But in strawberries, it's an octoploid. So octo, like an octopus, means that there's 8. So there's a lot of DNA for us to be able to extract here. You can try repeating this experiment with other fruits like bananas or raspberries or blackberries to expand your learning. But strawberries tend to yield the most amount of DNA. So if you have not already done so already, go ahead and take one of the components we need is isopropyl alcohol or rubbing alcohol. Place that in the freezer. We need that alcohol to be really cold, and we'll talk about why we need it to be cold in a little bit. But if you haven't done so already, put it in the freezer and we'll come back to it in a little bit, okay? So I'll give you guys a second to pop it in the freezer if you have not done so. As we're doing that, if we could go ahead and switch to my other camera, we'll start talking about what other materials we need. Okay, so again, our isopropyl alcohol is in our freezers. We'll come back to that in a little bit. So what do we need to conduct this experiment? What we need is, of course, our strawberries. And you can use fresh strawberries or frozen strawberries. Either one will work. 1 or 2 should be sufficient to be able to get a nice amount of DNA. We're going to need some water. Okay, so just some water we'll pull from there. Some soap, and we'll talk about why we need the soap. Some plastic cups or tubes. Some salt, and we'll talk about why we need this. Measuring devices, like a small spoon, tablespoon, or teaspoon. A measuring cup, mine just pops out. A stick of some sort, and this could be a coffee stir, a popsicle stick, a straw, anything that you have at home, something that's long enough to be able to grab our DNA. And a plastic bag. This is where our process is going to take place. And coffee filters, and we'll talk about why we need each of these components. Okay. So again, that isopropyl alcohol, your rubbing alcohol, if you haven't done so already, place that in the freezer, and we're going to come back to that shortly. Okay. Now, our first step is, well, we got to break up the strawberries. Okay. Remember, the DNA is embedded inside of these cells. It's inside the nucleus. And in fact, inside a single cell, there can be up to 9 feet of DNA. And when you eat an actual meal, you consume about 55 million cells. And so overall, you could be eating close to 90,000 miles of DNA, but it's so tightly wrapped in there. Or in South Africa, that's about 150 kilometers, or excuse me, 150,000 kilometers. So a lot of DNA. So we need to bust open the cells that are in here in order to get our DNA out. What we're going to be doing today, we're going to be doing a couple processes. We're going to be, again, extracting our DNA. We're going to isolate it, and we're going to be observing what's happening, okay? So take your strawberries. If you're using fresh, you can go ahead and just pull out the top, the little cap, the green part, place it to the side, like I'm doing as such. Just taking my caps off, okay? And go ahead and place these into your bag. And seal your bag up. Put those in there. And we're going to smash them. So if you like breaking things, this is your time to shine. So go ahead and just start mashing the strawberries up. Try to be gentle not to poke any holes in your bag, but we really want to start breaking down those cells. This is the first step in the process to start freeing up some of those cells so that we can be able to access it when we get ready to extract our DNA. So just go ahead and mash it up. It's going to take about a minute or 2 to start mashing it up. You can start seeing some juices coming out of there. So mash it, mash it, mash it. Get nice and finely pulverized, if you will. Again, try not to poke the bag. You don't want to create any holes. If you think that you're going to poke it, or when you suspect that you poked it, maybe place a second bag around it just to make sure everything is contained. We want to really get it nice and mashed up. Again, all we're doing here is we are freeing up some of those cells so that we can go in there and break up and extract the DNA. Okay, so again, I'm just mashing it up. Trying to break up all those big clumps. Okay, once you think that you've mashed it up pretty well, we're going to do a little bit more mashing in a second, but get that nice and mashed up. We'll be able to move into our next step. Okay, so again, I'm just working my fingers around it to smash all my strawberries. Again, this works with fresh or frozen, or if you don't have strawberries, try it with another fruit. Any type of mashable fruit, bananas, raspberries, blackberries, you can try this with. All right, so I have mashed up my strawberries, and I'm just gonna set this to the side, okay? Now, the next thing that we need to do is we have to actually make our extraction mixture. Now, this extraction mixture is really gonna work to break up parts of the cell so that we can free up the DNA. So DNA is soluble in water. So soluble, we talked about this a few weeks ago, just means it can dissolve in water. So we're going to need some water because that's what's going to be able to capture that DNA as we extract it from the cells. So go ahead and add about half a cup of water, or just over 100 milliliters, to another cup, to a plastic cup. So again, I'm just doing this, and we're just going to transfer this into a cup. Okay, so this is just half a cup of water. All right. Now, the next thing that we're going to add is some dish soap. And why are we adding washing soap? Well, inside of the cell, there's what's called a phospholipid bilayer, okay, that's enclosing it. Now, that phospholipid bilayer is actually insoluble in water, meaning it does not want to be around water. So much like when we have grease or something else on our hands and water just simply won't cut it, we use soap to be able to break down that grease and be able to take it away. The soap has the ability to form micelles and actually take and capture these more hydrophobic materials and help to remove them. And so we're going to use the soap to help remove and break down that phospholipid bilayer that's found in our cells. So we're going to need about 2 teaspoons, 2 of the small spoons of soap. So go ahead and get your soap, and we're going to place about 2 teaspoons into our cup. Okay. 1, got some bubbles. And number 2. Okay. We'll get it all in there. And then the last thing we're going to add to our extraction mixture is salt. And the reason we're going to use salt, well, that DNA is tightly bound with some proteins around it. And so this salt is going to help to punch some holes in those proteins and really open up that nuclear membrane to free up the DNA. So we're going to add 1 teaspoon, again, 1 of the small spoons of salt. So spilling a little bit, that's all good. So we have covers down. All right, and I'm just going to put that in there. Now, give your extraction mixture a good stir. We want to get all that stuff well blended. Try to avoid creating a lot of bubbles, but just give it a nice stir. Make a nice homogeneous solution. Remember, we learned the word homogeneous solution back on the 2nd week, which means that it's the same throughout. Everything is well blended. Okay, give it a nice stir. All right. And once you're satisfied that you've mixed that in well, we're now going to be transferring our extraction mixture into our bag with our strawberries. Again, the goal of the extraction mixture is to pull out and extract and isolate that DNA. We'll extract it, we'll isolate it later. So go ahead and pour it into your bag. Okay. And now again, before you seal it, try to squeeze out as much air as possible. That'll help make mashing it around a little bit easier. So I'm going to start closing it, then try to squeeze it, get rid of some of the water. So I may have a hole in my bag, so I'm going to add 1 more bag around this. Give me 1 second. So I'm just going to place this into a secondary bag to try to keep everything contained. In fact, you know what I might do instead is just pour it all in there to avoid it. I'm actually just going to pour mine in. If you got a hole like I did, pour it all into a new bag. There we go. And try to, again, make sure you get all the strawberries in there. Okay. We'll leave a little bit behind, that's all good. Okay. So again, I'm just going to seal this up. Try to get out some of that air. All right, and now I'm just going to take and work that extraction mixture around with that strawberry mixture. I really want to allow this extraction mixture to interact with those cells, start breaking down that phospholipid bilayer, breaking down that nuclear membrane, punching holes in the protein to free up that DNA. Okay? Again, because the DNA is soluble in water, it's going to end up being trapped or extracted into our water. Okay, so we're just going to mash it around. This is going to help open these cells up. Okay, really just working it in. Probably smells good. You can smell the strawberry scent as you're doing this. Okay, so a couple more seconds of allowing the extraction mixture to combine and work in breaking open these cells. All right, so at this point now, we've broken down our cells. We should have our DNA is now floating around in this extraction mixture. We now want to be able to isolate the DNA itself from this mixture that we have, this mixture of strawberry, some of the protein, the other stuff that's in there is now broken down and floating around in here, okay? And so this is where our coffee cup filters are going to come into play. So you can grab another cup and take one of your coffee filters, and you're going to have to use a little bit of engineering, if you will. Place your coffee filter into your cup. Put it down a little deep in there, but leave enough on top so you can kind of hold it, form this kind of seal. Okay, hopefully you can see that. Okay. Parents, if you're helping out, maybe you can hold it and allow your child to pour. But what we want to do is we want to capture some of this more solid material in our coffee filter and get our extraction mixture into the bottom of that cup. Okay. So I'm just going to slightly open my bag here. I'm going to, again, hold my coffee filter in place and begin pouring it into my cup. And we're going to allow a little bit of time. Don't try to pour it all in there at once. You got to let it start draining down. So you can see it hopefully coming into the cup. It's going to capture that solid material up top. So just take a moment. Again, don't try to pour it all in there at once. It will overflow. Okay, but you can start to see that strawberry, the extraction mixture is getting captured in that second cup. If you have a strainer, you could also use a strainer and instead of a coffee filter, help the mixture go in a little bit faster. If you're following along with us, feel free to show your cups so we can see how your extraction is going. And we're just letting this all drain out. Again, if you're just joining us, make sure that your rubbing alcohol was placed in the freezer so it can be nice and cold. Okay, again, I'm just working it around, trying to get as much of that liquid in there as possible. When you're filtering, it does take time because as more and more of the solid material is captured, it's going to take a little bit of time for the liquid to pass through the pore sizes. Okay, put a little bit more in there. Coming slow. Okay. All right, I'm almost ready to get this last little bit in. And then once I get the last little bit in, I'm going to actually pick up my filter to help squeeze it and get some more of that liquid out of there. All right. Okay, so now once I've got most of my liquid in there, I'm going to take, oh, don't be like me, it dropped. I may have to filter one more time. Give me one more cup, excuse me. What I wanted to do there was pick up my filter and squeeze it, but I dropped it. And that happens in science sometimes. So we just repeat. So new filter, just gonna transfer one more time. Let me double filter now. Again, we're filtering just to try to capture a lot of that strawberry, the solid material, because our DNA is not in our liquid. Just moving it back and forth a little bit just to try to speed it up just a little bit. You could also place like a rubber band around your filter to hold it in place. And we're going to get ready to grab that isopropyl alcohol in a second, but again, keep it in the freezer. We need it to be nice and cold to help isolate the DNA. There we go. That's what I was trying to do last time. I'm just squeezing it to try to get a good amount of that liquid in there. Okay, so that should be more than enough liquid to be able to extract and isolate that DNA. All right, so I'm just gonna place that to the side. All right, so what we're gonna need now is our isopropyl alcohol. Our DNA is in here, but right now, remember, it's soluble in the water, and so we can't see it. So we're gonna use this isopropyl alcohol, which has a different polarity than the water. That's going to now allow the DNA to precipitate out of solution, meaning it's going to crash out and we're going to be able to visibly see it. So we're going to use this ice cold isopropyl alcohol, rubbing alcohol, because again, the DNA is not soluble in there, and especially in these colder temperatures. So you want to avoid, when you're grabbing your isopropyl alcohol, avoid holding the bottle with your hands because your hands may warm it up. Okay, so try to hold it from the top. All right, I'm going to grab mine out of the freezer. Be right back. Okay, and we're going to need about 5 milliliters or so, or a couple teaspoons of your cold alcohol. Okay, so you can use your spoon again. Again, try to hold from the top of your container, whatever your container is in. And we're just going to take and add a few teaspoons of this nice cold alcohol into our mixture. Add 2, okay? And what you're gonna notice is that there's gonna be this layer formed at the top because you have these 2 different polarity compounds that are there. Now those white fibers, I don't know if you guys can see that white stuff that's starting to form. I'm actually gonna add just a little bit more just for the camera purposes. But again, you should be sufficient with 1 or 2. The white stuff that's starting to form in that layer, that is our DNA. And so if you look at it from the top, you can start seeing a bit of these fibers forming, or you may see this layer that's created. Our DNA is now in there. So to actually pull out our DNA and to be able to see it, this is where we need our sticks. So again, popsicle sticks, straw, a stirrer, anything will work here. So you just want to very gently, try not to dip into the bottom layer, but just swirl your stick around. I don't know if you guys can see that. Yep. Swirl your stick around in that top layer, and you can start grabbing your DNA. And so this here represents the DNA that was in those strawberries. So you can see there's a lot of it found in there. Remember, within a cell, you can have up to 9 feet of DNA. I don't know what the metric conversion would be off the top of my head, but there's a lot of DNA there, okay? And so in order to do this, what we had to do was we had to use that extraction mixture. So again, this is the DNA found right here at the bottom. If you had a microscope, preferably an electron microscope, something more powerful than what we used last week, you would be able to see more of the detailing that's here in this DNA, okay? But right now, we can't only see these long fibers, these long strands of DNA that we were able to pull. So again, what we've done here is we have extracted our DNA. We use our soap to actually break down the phospholipid bilayer. We use our water because DNA is soluble in water. We use salt to punch holes in the nuclear membrane and break down the protein. We broke the cells up, mashed them up, allowed that extraction mixture to interact with the cells, and then we filtered to be able to remove and isolate the DNA from the solid material. And then the last step is using this cold isopropyl alcohol to be able to pull out our DNA, okay? How did you all do? Were you guys able to get your DNA extracted? If you were, go ahead and hold it up. And if we can switch back to my other camera, please. All right, I see somebody. Nice, very nice. All right, can we switch back to my main camera? Awesome. So I hope you guys enjoyed that really quick demo using some common household supplies. We were able to very quickly extract DNA from our strawberries. Now I want to challenge you all to try this with other fruit. Think about what other fruits you could possibly do this with. Again, you have bananas, you have blackberries, raspberries, blueberries, anything that you can easily mash up, you can take it through this process and try to guess which ones you think will produce the most DNA. Again, strawberries are great because they have the octoploid of chromosomes, so there's a lot of chromosomes there, but the other fruits will work well too, all right? Any questions? Anybody? I see that people were able to pull out their DNA. Well, again, I do thank you all so much for joining me on this journey. It has been my pleasure to collaborate once again with SciFest Africa. SciFest Africa will always have a place in my heart. Ever since the first time I came there in 2015, I have enjoyed being able to interact with the learners and the educators and the parents that have come and supported this organization. So it has been my honor to work with you all in this virtual environment. I hope that you all learned. Again, if you've missed any of our previous sessions, definitely check out on the YouTube page. All of the recordings are posted. You can follow along with us. There will be some more virtual programming coming from SciFest, so definitely stay tuned. I wish everybody well, stay safe, and thank you so much for joining us with Steam Forward and SciFest Africa.
I've got a question.
You got a question? What's your question?
Is a human DNA, is a person DNA, is one person DNA the same as another person's DNA?
No, in fact, they are different. Unless you have twins, identical twins, that's one of the situations in which the DNA will be similar, will be the same. But no, people's DNA, and that's what, like in forensics, when we're trying to solve crimes, we use the fact that everyone's DNA is different to be able to tell people apart. Now, the interesting thing with identical twins, because they came from the same egg in this split, they do have the same DNA. So the best way to actually tell them apart is by using their fingerprints. So that's how you would tell identical twins apart. Okay.
Okay. Thank you. Thank you.
Thank you, and thank you guys so much for joining. I hope you all had fun. Continue learning, continue exploring science, STEM, and I will see you guys soon.
Thank you.
Thank you.
Bye, guys.
Thank you.
Thank you.